Okay, this is ALHS 1011, and I am going to share a file. Okay, let's back it up one. Okay, this is structure and function, ALHS 1011, and this is module 3. This is the second part of the nervous system on the senses. These are our standards, our KMS standards, and the learning objectives. The sensory system. Sensory system, basically, we spent the last chapter learning about input and output, motor and sensory neurons in the nervous system. This is really kind of honing in on the input from the environment, and as the brain can interpret the information being sent to it. So the nervous system has input, afferent, sensory neurons that brings in information in the nervous system, and the sensory system helps the brain understand and appreciate the experience. We have two different kinds of senses, special senses and general senses. Special senses are sight, hearing, balance, taste, and smell. You might not have thought of balance. General senses are just what they sound like. They're more general. They're not necessarily have their own specific organ. So thermoreceptors in the skin, especially, detect heat and cold. Mechanoreceptors, touch, vibrations, stretch, pressure. And nociceptors are pain receptors. Most of these are in the skin. And most of the special senses have their own organs. So as I mentioned, touch in the skin. Touch receptors in the skin are small round bodies called tactile corpuscles. We have a lot of those in our fingertips and also the tip of our tongue. And we get a lot of information from those corpuscles. We get sensations. So is it prickly? Is it soft? Is it cold, hot? All those kind of things. We get a location where exactly we feel something. The intensity, so low to high, and the duration. How long do you feel that for? And as you can see in the skin, we have nociceptors, which sense pain, thermoreceptors that do heat and cold. Right here, you can see that located in the skin. And I like this graphic. This kind of shows you, different things that you can feel. You can feel touch, you can feel tickle, pain, traction or pulling, heat, cold, pressure. So lots of different tactile sensations. As I mentioned, nociceptors are receptors that detect pain. And so when chemicals are released from damaged tissue, the nociceptors pick up on that. And if that is in the head, then cranial nerves are involved, sending the signal to the brain. If it's below the head, remember the spinal nerves are below the head, then the messages get sent to the spinal cord. So cranial nerves above the head, spinal nerves below the head. And we have two pathways where pain signals are sent to the parietal lobe. That's where they're sent, the parietal lobe. Remember, the frontal lobe is in front, then the parietal lobe directly behind it, and then the occipital lobe. So it's kind of the top of the head. One is a direct pathway, and the second way is a reticular formation in the brainstem, and then it sends it to the parietal lobe in addition to the hypothalamus and the limbic system. So a little more complicated. So the second way, not going directly to the parietal lobe, is it goes to the reticular formation in the brainstem. Remember that from the last chapter. And then from there, it goes to parietal lobe, hypothalamus, and limbic. And if it goes that way, we may get fear and nausea as a response to pain. There are different sensations of pain, and what we call referred pain means that it originates in one internal organ, but it is felt in another region of the skin. So for example, if you see on the diagram here, if you have liver or gallbladder pain, you may feel that in your neck on the right side. Remember, anatomical position, right side of your neck. Let's see what else is kind of a strange place to feel pain. Stomach is in the center, heart is right here and then down the arm. We kind of expect that. Heart can also be in the posterior side in the center of your back. So sometimes pain isn't felt exactly where you would imagine, right over the organ. Phantom pain, what does that mean? If someone has had an amputation, they no longer they can feel pain in the limb or the body part that is missing, that has been amputated. It's no longer existing, and that absolutely is real pain, and we medicate as such, and that's a very real phenomenon. Okay, moving right along, we're going to start with taste. Taste is also called the gustatory sense, and our primary organ for taste is our tongue. And on the tongue, you have lingual papillae. And the papillae are, here's kind of a blow up of them. Okay, some different shapes of papillae. Those are really have taste receptors on them. Sometimes we call those taste buds. Okay. We also have some taste buds in our lips and the back of our throat, but primarily those lingual papillae with the taste receptors and taste buds are on the tongue. The anatomy of one taste bud. So we have taste cells that have little taste hairs, microvilli, and chemoreceptors. So you have chemical receptors, and this would be your taste pore. There's a basal cell that they're kind of the stem cells in the taste buds, and then support cells giving that taste bud some structure. Okay, it's a support. In this case, it's called a supporting cell. So the sense of taste, we have 5 different possible tastes that we can detect. Sweet, sour, salty, bitter, and umami. And you might not be as familiar with umami, but it's a distinct taste of glutamites. Sometimes that's found in like soy sauce. In this case, you've got a little bottle of MSG down here. So it cannot be duplicated, and it's not a combination of the other 4 tastes. So I think previously they used to believe that there were 4 tastes possible, and then they realized this is its own unique taste. Taste preferences change with the body's needs. Pregnant women crave different foods through pregnancy. And the refinement of food taste is dependent on the sense of smell and the number of functioning taste buds. So we need good functioning taste buds and a good number of them, and also good sense of smell in order to taste. Cranial nerves and the tongue. We have a few that are involved here. Taste buds send signals directly to the brain through 3 different cranial nerves. One cranial nerve detects the anterior 2/3 of the tongue. So the front 2/3 is one cranial nerve. One cranial nerve detects the back or posterior third, and one for the throat. sense of smell. Your sense of smell arises from the olfactory mucosa in the upper part of your nasal cavity. So if you have your nose and your nasal cavity here, you have olfactory mucosa with olfactory cells and olfactory hairs working to operate your sense of smell. And these are bipolar neurons. You see the hairs here. We've got an olfactory bulb, an olfactory tract here with olfactory cells. And that is cranial nerve number 1. And this is the only sense that does not go through the thalamus on its way to the sensory area. The sensory area is in the temporal lobe. And then it gets interpreted, so the temporal lobe on the side, and then it gets interpreted through the front of the brain, frontal lobe. And we have a lot of physical and emotional reactions to smells. Especially when that sensation reaches the hypothalamus, the amygdala, emotion gets involved. You know, smells can bring us back, make us feel different things and that sort of stuff. So really the sense of smell, we're going to identify it in the temporal lobe, but interpret it in the frontal lobe, the thinking part. The next one is the ear and the sense of hearing. So the ear also is responsible for maintaining equilibrium or sense of balance. So we get 2 for the price of 1 with the ear. We get hearing and the sense of balance. And what hearing is basically is vibrations. So vibrations through the air come through the outer ear into the middle ear and become translated into nerve messages, and we interpret the sound. And that is the 8th cranial nerve that does that. It's the vestibular cochlear or auditory. The ear is separated into 3 parts: the outer, the middle, and the inner ear. The middle ear contains the tympanic membrane and the tympanic cavity. And the inner ear is more the labyrinth or the maze of the cochlea. Here's a good picture of this. So we have the outer ear, which is the auricle, the lobe, the parts that we pierce, right? We have the external auditory canal. Then the middle ear really starts with that tympanic membrane, or also called the eardrum. And these little bones called the malleus, incus, and stapes. And then the inner ear, we have, it's also known as a labyrinth, the cochlea, which looks like a wound up shell, the vestibular nerve, sound window, some semicircular canals. And there's fluid in here, and the vibrations will get interpreted to the brain through the auditory nerve. We also have a pathway to the pharynx. The external ear, the outer projection, we call it this the pinnacle, or the pinna, or the auricle. Pinna or the auricle. We also, inside this first part, we call the auditory canal, or the external auditory meatus. Meatus just means opening to the outside. In the middle ear, we have, it's also known as the tympanic cavity. We have those 3 little bones I mentioned before. Those 3 little bones are also called ossicles. And the ossicles are joined and they amplify the sound. The tympanic membrane receives the sound, and these ossicles, these little tiny bones, amplify the sound. Once they're amplified, then the sound waves are transmitted to the inner ear and make vibrations in the fluid in the inner ear. The eustachian tubes allow air pressure on either side of the eardrum to be equalized. So not to, you've been on an airplane and you have your ears kind of popping. That's built up pressure, releasing pressure. You want the air pressure on either side of the eardrum to be equalized. And there are tubes that connect the nasal cavity to the pharynx and the middle ear. So if you can see over here, yes, here, the tympanic membrane, the external auditory canal connects to the middle ear, the inner ear, but also the pharynx. Okay, here's a little, I was kind of trying to explain why do your ears pop with pressure changes?
It's time for today's Did You Know? Now, anyone who's ever flown in a plane has dealt with their ears popping. But why does that happen? Is it A, the airplane is moving up too fast? B, there's a mismatch in air pressure? Or C, your eardrums are inflating? Before the break, we asked if you knew why your ears pop when you fly in an airplane. And now it's time for the answer.
Your ears pop in an airplane because of your eustachian tubes, the tubes that connect the space underneath your eardrums to the back of your throat. You're getting a cabin air pressure mismatch with your middle ear pressure mismatch. When we go up in a plane, the cabin pressure of the plane is changing, and suddenly the pocket of air underneath our eardrum is either too small or too large, and it pushes our eardrum out or it sucks our eardrum down. What that popping is doing is that's forcing air up your eustachian tubes underneath your eardrum, and it's allowing your middle ear pressure to equilibrate to the cabin ear pressure, and then suddenly your ears feel better.
Okay. So these are the ossicles I mentioned. These are the three little bones in the middle ear, and they're kind of named for the shape that they have. So the hammer, or the malleus, looks like a hammer, is attached to the tympanic membrane. That's the first one. The second one is the incus attached to the hammer. The third one is the stapes, or stirrup, that is attached to the oval window, a membrane that brings it to the inner ear. So you have the oval window. The oval window transmits the sound waves into the inner ear. Okay. The inner ear is divided into, we call this the labyrinth, the bony labyrinth. It also looks like a maze. It's got some hollow spaces. And we have three kind of areas of the inner ear: the vestibular chamber, the semicircular canals, this kind of spiral things on top, and then the cochlea, which looks like a snail shell. The cochlea is that snail shell looking thing. It's a bony spiral shaped entrance to the inner ear, and it connects the oval window membrane. There's fluid in the cochlea called perilymph, and when sounds are amplified, that fluid helps transmit sound by vibration. So that endolymph, that fluid in the cochlea, carries the sound to little tiny hair-like receptors. Remember in the nose, we had little hair-like receptors, same thing here. And those hair-like receptors get stimulated and then conduct a signal to the brain via the 8th cranial nerve, which is the vestibular cochlear nerve. So here's putting it kind of all together, how it works. Sound waves enter the external canal, vibrate the tympanic membrane in a process that we call sound conduction. So we hear something, sound. It goes to the tympanic membrane, or the eardrum. And then the middle ear is responsible, remember those ossicles, the malleus, the stapes, and the incus, those are responsible for amplifying that sound. So we go from sound conduction then to bone conduction. Those stapes cause like a gentle pumping against that oval window membrane, and the cochlear fluid begins to vibrate. Small little hair-like neurons found in an area called the organ of Corti. So then those movement of the hair neurons and the movement of the fluid causes a nerve impulse, which gets sent to the temporal lobe of the brain, which gets then interpreted in a process called sensory neural conduction. So sensory neural conduction. So we've changed something like sound to bone conduction, fluid movement, hair movement. Now it's nervous system. So outer ear, we got our sound waves. Outer ear, the auricle, goes through the external auditory canal, vibrates that eardrum, that tympanic membrane, and that three little bones, the ossicles, vibrate and amplify the sound, which causes the perilymph, the endolymph in the cochlea, the fluid, to move and kind of stimulate in that oval window right here, to stimulate the hairs and the neurons in the auditory nerves that goes to the brain. Okay, so I mentioned that the ear also has to do with vestibular or balance. And there are two different types of equilibrium. We have static, where the head is stationary or moving in a straight line. And we have dynamic, where the head is rotating. And we have equilibrium receptors in what's called a vestibular apparatus in the ear. And the vestibule of the inner ear, you've got for static and equilibrium, that was the head either stationary or moving in a straight line. The saccule perceives vertical movements of the head. The utricle perceives horizontal movements of the head. The macule is a patch of hair on the saccule and the utricle. You have the autolymphatic membrane, a gel-like structure on the macules. Otoliths are what we know as ear stones, and they move and bend the hair cells with gravity. So that's what causes us to realize that we're balanced or not balanced. And then dynamic are sensed in the semicircular canals. We've got crista ambularis, which is a patch of hair on these canals. And then we have some endolymph, the fluid that flows in the semicircular canals and bends hair cells. And all of this gives us our sense of balance. You do not need to know all these parts to the vestibule, just this general sense that static, what is dynamic, and what is happening here in the inner ear. Okay, moving right along to the eye. The eye is similar to an older type film camera, if you're old enough to remember that. Light comes in from an image, it passes through the pupil, through the pupil, through the lens, and gets focused on the back of the eye on the retina in what we call photoreceptors or light receptors. The iris, the colored part of the eye, allows the right amount of light to enter the eye for proper focusing. So the iris is going to control the amount of light. On the eyes, we have eyelids and eyelashes, and the eyelids close over the eye to protect it from light, particles, or injury. The eyelashes, again, help prevent large particles from entering the eye. The eyelids contain sebaceous glands, oil on the eyelids. And they keep the eyelids soft and pliant and make the eyelashes a sticky trap. So that oil produced on the eyelids kind of coats the eyelashes and causes it to trap any particles that would be harmful to your eye. Your external structures of the eye, you have the orbit, which is the cone-shaped cavity where the eye is housed. It protects the eyeball. This cavity, the orbit, has fatty tissue to prevent it and cushion that eye. Then we have 6 short muscles that connect to the eyeball and allow rotary movements of the eye. Okay, also within the eye is what we call the lacrimal apparatus that produces, stores tears. It has the lacrimal gland, ducts, and a lacrimal opening. The lacrimal gland produces tears, and when we blink, that cleanses and lubricates the eye. And our tears have like antiseptic properties. It prevents an infection of the eye. The conjunctiva is a mucous membrane, the covering over the surface of the eyeball. It's a protective covering for the exposed part of the eyelid, the surface of the eye and the eyelid. So the conjunctiva is not covering the back part of the eye where there's no external exposure, only underneath the eyelid and the exposed part of the eye, surface of the eye. Here's a good picture of the internal structures of the eye. The eye is globe-shaped and it has 2 chambers of fluid. The vitreous humor, the main part, and the aqueous humor in the front. Okay, so talking about the humors of the eye, the aqueous humor, like I mentioned before, in the front of the eye is watery and it bathes the iris, it bathes the pupil, the lens, and it fills both the anterior and posterior chambers. The vitreous humor in the vitreous chamber, or the most big part of the eye, is more jelly-like, less watery, but clear, and it occupies the entire cavity behind the lens. We have 3 layers to the eye. We have the sclera, we have the cornea, and we have the retina. So here's sclera is the white part. Then you have choroid, you have the retina. Here's another one of the labels of the eye. So the sclera is the white part of the eye. The conjunctiva is the clear part protecting the exposed part of the eye. The sclera, the white part of the eye, is tough fibrous tissue and it's protective. Part of the cornea is, or part of this, the sclera is the cornea. And that is transparent. It allows light to enter the eye, pass through, and get curved and focus on the retina. So the cornea is very, very important and has 6 layers. So we talked about the sclera, the white part of the eye that contains the cornea. Now the choroid. The choroid is the next layer. It's highly vascularized, it's pigmented, it's that middle layer providing nutrients and nourishment and blood flow to the eye. It contains the iris, which I mentioned before is the colored part that controls the opening. The opening is the pupil, and the pupil is where light passes into the eye. The colored part, the iris, has a muscular sphincter that if it's in low light, it relaxes and widens. And if it's in high light or bright light, it constricts. So it controls more or less light. The lens is directly behind the pupil, and it is, it looks like this, the blue diagram, and it is surrounded by ciliary muscles. And these muscles will shape or morph the lens, making it thinner or thicker to allow incoming light to focus. We want the focus of the light to end up on the retina, and that is done possible through the lens. And what we call that is accommodation. And basically, the pupil changes, the lens curvature changes, and it makes sure that the image converges in the same place on the retina and gets properly focused. Okay, so I mentioned this retina a lot, and this is very, very important. The third layer, so we have the sclera, the choroid, and the retina. The third layer is a delicate membrane. It's the back of the eye, and it has two types of photoreceptors or light sensing receptors called rods and cones. Rods are active in dim light and do not detect color, where cones are active in bright light and do perceive color. The receptors have a chemical change. They change color when in the photopigments when light hits them. And those impulses, when light hits the rods and cones, and there's chemical change that gets converted into a impulse that gets sent to the optic nerve. And the optic nerve is what goes to the brain. So the information gets sent from the retina, the rods and cones, the chemical change happens, that impulse gets generated, and the impulse goes down the optic nerve to the area in the back of the head, the occipital lobe, called the visual cortex. In the back of the cerebrum is the occipital lobe. So again, let's put this together. We have light rays. Okay, light rays pass through the cornea, which was the transparent part of the contained on the sclera. It goes through the aqueous humor, that front fluid, then the pupil. The pupil may change size by the ciliary muscles. Also, the lens may accommodate, and ultimately it goes through the vitreous humor in the center of the eye and gets focused, number 4, on the retina. On the retina, you have photoreceptors, so light receptors, called rods and cones, which causes a chemical change, which causes a nervous system change. And that nerve impulse gets sent down, electrical impulse gets sent down the optic nerve or cranial nerve 1 to the occipital lobe, or I'm sorry, cranial nerve 2 to the occipital lobe of the brain. And that's where it gets interpreted as vision. Okay, so there's a few cranial nerves involved. The optic nerve, as we mentioned, coming off the eye. The ocular motor nerve, cranial nerve number 3, and that has to do with eye movement, controlling the lens and the pupil, constriction and dilation. And then the trochlear nerve, cranial nerve number 4, has to do with eyes moving down or side to side. We've got more. So cranial nerve 6 has to do with turning your eyes outward. Trigeminal nerve carries sensory information from the upper eyelid to the eye muscles. And there are different ways to assess these nerves. And the facial nerve can also control the lacrimal glands. So 6 different cranial nerves involved in the eyes and vision. Okay, I think I just double checking, but I think we are done. Yes, the grayed out Slides are just additional information if you need more resources.